Thank you, everybody, for taking into the presentation today. And we are very pleased to have our speaker, Charlotte Piette, providing a lecture for us. So some background for Charlotte is that she got a PhD from the College of France in Paris. In her PhD, she was actually co-advised by her own Jonathan. She is currently a postdoctoral researcher in Bob Data's lab at Harvard. Some of her research interests are in math and neuroscience, including learning and memory. She has worked in areas such as engrams, the visual system, and synaptic plasticity. So we very much look forward to hearing about her endocannabinoid and learning research today. So.
Hello everyone. Thank you for the invitation. And so yeah, today I will present you a work that I explored during my PhD in Paris, and that will hopefully be published soon. And so the project started with the aim of kind of bridging a gap between a plasticity that had been described in vitro, so at cortical striatal synapses by the Valence lab, so in Paris, and its possible function in vivo. And so due to the features of the plasticity that I will kind of describe a little bit more in details later, like we hypothesized that this plasticity could be involved in, one-shot learning. And so I'll give you a brief introduction on this one-shot learning kind of idea, and then move to the features of this plasticity. And so an intuitive definition of one-shot learning is that it's basically that a single experience is sufficient to induce a long-lasting memory. And we can think of many situations actually like that rely on such, that can rely on such one-shot learning process. Like if you think of a face, for example, that can, be remembered, or at least recall after just a very brief encounter. Same goes for sometimes even more complex situations, usually like some emotional kind of saliency to it, and that becomes part of our episodic memory. In the case of animals, we can think also of situations that involve quite a strong survival value. Like if you encounter a predator, then you will try to avoid the territory associated with it, or you would actually, the opposite, like try to remember like a food source or water source that you discovered once. And what I also find quite interesting is that at a more conceptual and like cognitive level, you can also learn like a new concept or a new word just after a single presentation. And so I chose those examples also to highlight that we can also think of adding a kind of second feature to the definition of one-shot learning. So it's not like necessary or required, but it would be meaningful in the later part of the talk. And so the experience can be unique, but it can also be extremely brief, and that can be actually sufficient to trigger a long-lasting memory. So in those examples, like a few seconds can already be sufficient to form a memory. And so based on this, like even though we can expect that many different brain areas could be involved, like in those different scenarios, I think like a common constraint that comes from that like brevity is that just a few spikes or bursts of spikes might actually be enough to produce that long-lasting memory trace. And so this idea that few spikes or few bursts are enough is also kind of backed up by some evidence that actually during like a short sensory stimulation, or like during novel object presentation, that there are actually few spikes happening. And so there are like some recordings that have been validating this kind of assumption. And again, if you have questions, feel free to interrupt. And so if we assume that a few spikes can be enough, then the question is, how can this be implemented concretely, like at the synaptic level? What kind of plasticity rules like kind of match this criteria, and how can we also read out those spikes from like the background kind of ongoing activity. And so this question has somehow been investigated initially in brain slices, so ex vivo. So by conducting experiments basically in which just a one shot or like very few electrical stimulations were applied, and then long-term kind of plasticity changes were monitored, and those experiments were typically done at in the hippocampus, so at say 3, say 1 synapses, or also between cortical pyramidal cells. And so an example of this, for example, is that just a single burst, like a single stimulation here, was sufficient to actually drive like a long-term depression that you can actually record like over at least like an hour, like and so those traces will come back, but we basically record like the postsynaptic response of the neuron. So it's like the excitatory, like the postsynaptic excitatory potential. And another important rule that has been like discovered recently is behavioral timescale plasticity rule in the hippocampus, which also rely on just a single kind of stimulation. And like the one that I'm actually going to detail a bit more is this spike timing dependent plasticity paradigm, which involves basically like the stimulation of both pre and postsynaptic elements. And well, usually like those protocols rely on like a lot of stimulation, like more like 100 or like 200 stimulations. What the PhD lab in Paris found was that actually like 15, like 10 to 15 stimulations were actually enough to trigger some long-term plasticity. So if we focus a little bit more on that like plasticity rule, so again, so the protocol itself is that we pair like a pre and post synaptic elements. So in our case, it's like cortical striatal synapses. So we stimulate the cortex and we also like induce some spiking activity in post synaptic target, which in my case is like a single like a striatal neuron. So to record like long-term plasticity, what we do is to typically have like a relatively like stable baseline that you can look over here. So it's like the response to a presynaptic like stimulation, like again, like postsynaptic excitatory potential. And then after inducing that plasticity protocol, which are like those paired like stimulations, so close in time, we observe or not changes in the synaptic weight. And so here we observe like a long-term potentiation, like an increase in the response. And so that plasticity is like, to go quite quickly, was quite unconventional because it depended on a system which is like the endocannabinoid system, which is usually more associated to long-term depression. But and more intriguing is that, in fact, those synapses are capable of like responding to different protocols. And so if we change the number of pairings, actually, like the number of stimulation, what we observe is that there is a window of plasticity that is quite like classical, let's say, like the one that can be induced with like 100 pairings. And this one is actually mediated by a different signaling pathway, the NMDA. Then we lose it if we decrease that number of pairings, and then we uncover another like small pocket of plasticity, which is this endocannabinoid-mediated one. So we have very like a dual kind of signaling pathway here at the same synapse that can respond to either brief and like relatively light stimulation, and also a much stronger like repeated kind of stimulation. So which makes it quite particular, yeah.
May just be lacking some of the background here, but is it important that in this plastic response, the synapses getting depolarized versus like hypopolarized is the direction that the voltage is going in before and after the spikes? Is that important?
What do you mean by like
Is it
depolarize?
that the voltage is becoming less negative rather than more negative.
So we always depolarize the neuron when we do the stimulation itself. So to induce actually some excitability and some calcium influx. So that's kind of I'm not
Getting it closer to the threshold somehow.
So-
Because I think the point is that the synaptic wave is changing.
But
So
yeah,
in response, you're looking at the top. Yeah. So you're generating the same stimulation and you're looking at the effect on the nerve. So what you're looking at is the synaptic wave. So the plasticity is a change in the synapse.
yeah, not the
That's supposed to be long term and to store them. That's
Can you just explain on the x-axis? I'm a little confused because the y-axis is the output. You kind of, on the right side, the input
Yeah.
is post-pre versus pre-post.
yeah.
And is the x-axis like a constant frequency?
So the x-axis, yeah.
Or is it like 100 pairings? So the frequency is fast.
So the frequency here is left the same. It's like 1 Hertz, but it's really just the number of stimulation that we change. And so it's either 100, like 50 or 0, like or 5, like but so and then the post-pre and pre-post are like small variations in like which a neuron basically or like which area like is like triggered kind of first in the protocol, whether or not like
So I guess I'm asking with the ECB plasticity,
yes.
when you do 50 or 100 pairings, are you only getting depression if you switch the order? But if you kept it post-pre, you still
Yeah.
get potentiation? I don't know.
So yeah, like the potentiation that we see with the ECB with that is endocannabinoid mediated is only seen when you do post-pre. So first like the cortex being stimulated and then the striatal like response. And the pre-post is only like going to mediate LTD, but for like relatively strong stimulation.
I guess what I'm saying, so if you do
Yeah.
100 pairings.
Yeah,
post-
here.
Pre before, wait a second.
Post
This is split from cortex only, but.
Yeah, it's
but if you do 100
anti-Hebbian.
pairings post before pre, what is the result?
It's like an NMDA-mediated LTP.
Oh, okay, so but if you block that in the end, it's
There is no plasticity.
just be zero. wouldn't it?
Yeah.
So with the ECB, is there no mechanism like, because when you do this pairing and use calcium.
Yeah.
The same change in calcium is giving potentiation, but then also pressure.
So it's actually like the like model that was associated to the ex vivo experiment suggested that there was actually a difference in the calcium influx that was like induced by this stimulation, and so it was actually stronger when you were doing post-pre and like for a small number of pairings. Because you were also adding basically like calcium influx from the endoplasmic reticulum, for example, that get like kind of depleted for a larger number of pairings.
So like EER is actually.
Yeah, exactly. So you don't have that additional source, and yeah.
Back to the-
So yeah. Is that a bit clearer? So like 2 systems basically for mediating LTP, one for light stimulation, one for stronger stimulation. That's kind of what's important, I would say, to keep in mind at this stage. But yeah. And so kind of to come back to the working hypothesis of like the project. So if we assume again, like in a very like simplified way that synaptic plasticity is required for learning memory expression, so without necessarily assuming that it's actually where like the memory is stored, but what we want to test is that this endocannabinoid like mediated plasticity that we have been described in vitro could at least participate to the learning of a one-shot experience, so in vivo. Yeah, so that's kind of the main diagram, and so we've kind of been trying to test this hypothesis in through a series of experiments, and so of course it's kind of difficult to interpret each individual experiment that I will described, but I think like the kind of strengths come maybe from the fact of kind of the coherence, I would say, between all of them. So we'll kind of go through them. And so I'll start with a presentation of the one-shot learning test, which we use to kind of like ground the the study. And so the idea was to develop a test in which the duration of the one-shot experience could also be somehow varied, but without really being imposed or constrained by the experimentalist, and somehow making it also quite naturalistic for the mice. And so that involved a sticky tape. And so basically, the idea is that the sticky tape avoidant test engages the mouse with a loose piece of adhesive tape, which is placed like in the arena. And so the mouse very rapidly, like on the first exposure, will get stuck and kind of glued to it. Just one time, and usually very briefly, because it's able to remove it. And the idea is that on the next exposure, because it's a rather uncomfortable stimulus, it will avoid it. And so the protocol really starts with first an habituation session, so empty arena. And then so at the familiarization step comes this first encounter that I just described. And so what is interesting is that for 50% of the mice, approximately, like the duration of contact will last less than 15 seconds. So we're really like at the very one-shot kind of learning experience that I was kind of mentioning at the beginning, very brief. And so this session really is the one-shot kind of encoding experience. And then what happens next is the retrieval session, where we actually test the memory and the learning performance. And we can do this, for example, 24 hours later. And in this example, so I've just plotted the trajectory of the mice until the time of contact. And you can already see that there is like some avoidance and detours close to the tape in the sticky tape group. And we have also a control group, which is like, just a non-sticky tape like sitting in the arena. And so just to make maybe the task a bit more clear, like I have a video, so I don't know if it will play, but so we'll see like the beginning of the familiarization session. So I just put the mouse in. So the tape is usually placed also near the walls, because that's where the mouse typically likes to go. And so it's kind of removing that tape relatively fast. And then I will remove the tape from the arena. And then 24 hours later, same mouse. And now you see like those detours very obvious, like when it's approaching the tape. So you would think that, like something happened like that really make this behavioral change in this animal. And so there are like some relatively easy ways to quantify this behavior. And if we look, for example, at the number of detours or the latency before contacting the tape between like the familiarization and the retrieval session, we can see like on average that there is like an increase in the sticky tape group compared to the non-sticky tape. But again, like it's not necessarily that easy at the individual mouse level sometimes to actually like make sure like and have a clear like idea of whether or not it has learned the task. And so we also kind of included more parameters to make this a little less, to actually have like an index for each animal. And so this included like different parameters, like again, including the number of detours, but also like where like the mouse would like glue to the tape again on the retrieval session and so on. And so to see if we could actually get a more kind of a better quantification for each animal. And so this included some like like principal component analysis, and then like the definition of an index. And so like the idea here is that if this avoidance index is like larger than 0, like we can say that the mouse is basically like would be considered as a learner, like showing like this type of avoidance behavior that you saw in the video. And so yeah.
Can you say a bit about what went into the PCA? Like
Yeah.
What was the initial?
So yeah, so there were 9 parameters basically that we could quantify in a kind of relatively unbiased way, like across all the animals. And so again, like the fact that the latency to contact can vary a lot, we couldn't include like many other obvious, let's say, parameters, like the time that it would spend, like, you know, near the wall and the opposite, like outside of the arena. So we focused more on like the number of detours, like the side passages near the tape, for example, like whether or not it was like making very clear, like U-turns basically. And we count those. Yeah. Exactly. So we had like, yeah, those 9 parameters that we could just quantify very straightforwardly. And so yeah, and so then if we look at this avoidance index as a function of the duration of contact, well, so first, again, as I said at the beginning, we have quite a large heterogeneity of behaviors, even at familiarization. And so that's kind of what you observe here, like with the contact duration on the x-axis, like varying from like a few seconds to almost like the 10 minutes in some mice, like that, not even able to remove the tape so easily. But then if you look at the color code, well, what you observe is that basically like the distribution is rather uniform across that panel. And so the avoidance index doesn't really vary as a function of the duration of contact. And so in here, like the x-axis, I should mention, is like the time to actually actively remove the tape. So it's another kind of relatively correlated measure of the time spent with the tape. But as you can see, like on this little histogram, like there is not much bias between the contact duration and the proportion of mice really avoiding the tape at the end. So, and so really what is quite striking again is that even like 2 or 3 seconds of contact with the tape is sufficient to have like this long-lasting memory and avoidance behavior. And another aspect that we tested with this task is that what is the persistence of the memory, basically. And so we increased the like intervals between the familiarization and the retrieval sessions. And so I presented to you like right now, like until now, like the retrieval at 24 hours, but we tested like intervals like from 72 hours to like 1 month. And what we found is that actually like the percentage of mice and again, like the avoidance index associated to it did not really vary across those different intervals. And in addition to that, we also tested the robustness of the memory to like interference, let's say, like by adding some extra tasks that also somehow depend on the same kind of circuit, like the striatal circuit, by having the like mice performing like novel object recognition, accelerating rotor road, and like a kind of T-maze version, like cross-maze version. And same here, we didn't see much of a change in the performance on the sticky tape. So again, showing that this one-shot learning is very persistent and quite robust over time. And so now that we have this this task at hand, like the natural question is like, what kind of what's happening basically at those corticostriatal synapses? And so the in vitro plasticity that I described before has first been described between the secondary somatosensory cortex and the dorsolateral striatum, which is like one compartment in the striatum. And so we kind of focused on those 2 regions as well in vivo. And so to probe plasticity in vivo, we developed an approach that had been used by a previous paper, so which consists basically of recording like the excitatory response to some optogenically evoked like presynaptic cortical stimulation. So what we do is that we express the channel rhodopsin, which activates like cortical terminals in the somatosensory cortex, so in the presynaptic part. And we implant like an optrode, like so optic fiber and like an electrical wire attached together to record actually like the response of the striatal neuron to that stimulation. And so the response looks like this trace here. Oops, where is my here? So that like different components, but basically like the first phase would correspond to the activation of the channel rhodopsin, typically like the presynaptic part, and then the second phase would correspond to the excitatory like postsynaptic response of the striatal neurons. And so we kind of tried to monitor changes in the response of that second component over time, so at different stages during the task. And so here's an example of traces that is recorded from one animal, like across the different stages. And so we record like the changes in amplitude of that second component when it's like normalized again to the first amplitude. But so the expression of the synaptic plasticity basically is kind of like the response of that, the amplitude of that response here is like a proxy for the expression of synaptic plasticity. And so we did this in like 3 different groups of animals. And what we found is that there was no overall changes in mice that experienced just an empty arena or a non-sticky tape 24 hours after the encoding stage, like after familiarization. But there was like a significant LTP on average, significant potentiation on average in the sticky tape group. But as you can see with the like big heterogeneity across animals, like with some expressing like a clear LTP and others no apparent changes.
So you're activating a whole bunch vestry, like
Yeah.
vestry, and averaging the signal across the whole of this polystriatum.
Yeah, it's a very bulk
So
signal.
maybe no wonder, right? Because there are like theories that when you potentiate some synapses, you get heterosynaptic depression, weakened because you really want to get specific response. So the potentiated synapses could be very strongly increasing, but it's kind of washed out by the depressed ones and the non-changing ones.
So yeah, it's a very bulk signal, I would say, but the fact that we see it in I mean, we found like some correlates of this heterogeneity, so that we didn't really like correlate, for example, with the location of the electrode, for example, or things like that. So I'm not sure like why we would just see it in like a group of animals, right? Like so. But I would love to also have like cleaner kind of and more spatial kind of representation of these actually like signals, like that could also be useful. And so to kind of make sense of this heterogeneity, we went back to the behavior of these mice at familiarization and also at retrieval, tried to correlate it with some parameters here. And so what we found is that basically 2 main parameters were 2 main factors could actually cluster the group that expressed like long-term potentiation. So here on this axis, you have the contact duration on the x-axis, and then on the y-axis, the avoidance index, so at retrieval, so whether or not the mice have learned, so positive, meaning one-shot learners, basically. And then the color represents like the level of potentiation. So what you observe here is that there is a majority of like hot red spots basically in the brief contact one-shot learners, right? And much more like heterogeneous and rather no plasticity, even like some depression for the long contact duration. For the long contact duration, yeah. So we haven't, like we've been trying to investigate a little bit more that question of what happens for the long contact. We haven't have like a clear idea yet, but it could be that also other regions become involved and that some transfer also happen, or maybe that just a smaller actually number of synapses get really involved at that stage, like more specific, and so that we don't capture it necessarily. But yeah, what we really focus on next are like those this cluster of short contacts and one-shot learners. And so to kind of then approach the nature of the plasticity that we see in vivo in this group, like we decided to take advantage of a protocol that has been used ex vivo, so which is described as an occlusion or saturation phenomenon. So basically, like if an LTP has already occurred and has been already expressed at some synapses, typically because of like learning or like some experience like that the animal has encountered, it will kind of ultimately limit the ability of those synapses to further induce like potentiation or LTP. And this saturation is supposed to be pathway dependent. So if it's an eCB-mediated LTP, we shouldn't be saturating like an NMDA-mediated LTP. And so with this logic, we thought that we could actually distinguish ex vivo whether or not an NMDA versus eCB-LTP plasticity had been occurring during this one-shot experience. And so for that, we basically cut brain slices. So this time not from a naive animal, but from mice that have experienced this one-shot exposure with a tape. And we applied the same kind of protocol that I described before, like those 15 kind of pairings, spike time dependent plasticity, to investigate either like the eCB-LTP pathway or like the 100 pairings to investigate the NMDA LTP pathway. So we first had like some controls, so which are the no tape group. And in this group of mice, we could evoke both the eCB-LTP. So as you see, like there is like some potentiation being expressed, and also for both the eCB and the NMDA protocols. So which is kind of consistent with the absence of LTP that we observed in vivo, right? And then for mice that experience the sticky tape, so we did the protocol either 30 minutes post-familiarization or 24 hours. And so here in red, so you can see mice that are short contact with the tape. And so what you can see that in both cases at 30 minutes or 24 hours, there is basically on average like no plasticity that is actually evoked, right? Like the curve is basically flat. So supporting the idea that the eCB pathway is somehow occluded at those two time points. And if you compare it to the long contact group, which is in green, like it's actually quite reverse, right? Like the long contact group still express the eCB-LTP. And in the case of the NMDA component, we also try to take a look at this. It's a little more variable, actually, like if you look at the individual data points, but on average, like what we observe is that there is still actually some LTP being observed, like especially like if you look at the 24-hour group, like it's more, it's actually more clear, like in the short contact. So the NMDA pathway doesn't seem to be really involved in this task for the brief contact. It's seems the opposite for the long contact. So quite a reverse kind of situation here, like between short and long contacts and eCB versus NMDA pathway. And again, which makes sense if you think again of how those two pathways are evoked in vitro, right? Like the eCB is evoked in like a light stimulation, so maybe a short contact, right? Like if we make a parallel versus the NMDA, like repetition, like longer contacts. So that's kind of the idea. Yeah, so those are actually like correlation that we observed with the duration of contact between the two occlusions. And an additional thing that we also checked is whether or not there was also some link between the learning and the occlusion. And so this time, like the mice have actually like been exposed twice to the tape, right? To actually be able to assess the learning performance. So we do the occlusion right after, we do the protocols right after like the retrieval session. And what we found is that actually like the mice that have learned the task compared to those that didn't learn show more of the occlusion of the eCB-LTP pathway. So it's actually highlighted here. So it's kind of the reverse picture. If you remember the in vivo like diagram, with this time like occlusion being like in the cold like blue colors or like white, found for the one-shot learners in brief contacts. So yeah, and so we're very quickly also to further consolidate the eCB hypothesis and test whether eCB transients were actually evoked in vivo during the test. We also measured the eCB release during the contact with the tape. And so we used for that some fiber photometry recordings, so in mice that expressed like a sensor for endocannabinoids. And we found that there was actually an increase in the fluorescence, so increase in the release of endocannabinoids. So after the contact with the tape, that lasted about like 10 seconds. And we mostly had like in this case like short contact. So I don't really have the long contact situation. So yeah, and so kind of the next step now is to get a more causal kind of manipulation, let's say, approach in which we prevent the expression of the eCB-LTP and test whether learning of the sticky tape is impacted or not. And so for that, we used two approaches, and the first one relies on transgenic mouse models. And so those models basically have, so we used two different ones. Either they no longer express the CB1 receptor, which we know is kind of critical to mediate this eCB-LTP. And the second one is a D2 knockout, but specifically, again, like targeting some receptors that are expressed on the presynaptic terminals, which we also know from like the in vitro characterization of the plasticity are kind of essential for the expression of the eCB-LTP. And so we to make sure the specificity again of the receptors that we block, that we no longer express, we do like viral injections, and then we kind of want to first validate that the eCB-LTP is no longer expressed. So these are recordings that we did in vitro, like in naive animals, and then we want to check the behavior of those animals on the sticky tape task. And so that's what I did next. And so again, it would be kind of always the same graph, right? With the contact duration on the x-axis, the avoidance index on the y-axis. And so here the Cree animals are in red, so the knockout, and the control are in blue. And so if you look again at this, you focus on the top left quadrant, like what you see is that there are basically almost no red dots, right? Like what you would expect to actually see some in both cases. So meaning that basically there are no one-shot learners for the knockout animals when they are exposed to a brief contact with the tape. And so this is also quantified over here. Like if the contact is like shorter than 20 seconds, you basically have like an impairment in the one-shot learning, which is not the case when the contact is longer, like where at least like you recover, especially more so for the D2 LOXP, knockout kind of a normal learning curve. And the effect is quite strong, as you can see. And interestingly, another feature of these knockout mice is that they are actually still able to learn the task, not just after one shot, but after 2 shots, basically. So if we let them be exposed to the tape a second time, so and test the mice on a second retrieval session, we can see that they actually improve their performance and kind of reach a relatively similar level to the wild type, to the control mice. So it really means that the eCB-LTP seems to be engaged only, to really be required to drive learning just at that very first unique experience moment, kind of, and other plasticity mechanisms, take over or compensate when the stimulus becomes kind of repeated. And quite rapidly, like we also tried to explore this also manipulation approach by using a different a method, like pharmacology this time, to block more transiently the receptors. And so that enables us to better target the learning part and disambiguate it from the memory expression or memory retrieval process, let's say. And so we did this by targeting either CB1 on the NMDA receptors, specifically in the striatum by using cannula, where you can basically inject drugs specifically in these areas of the brain. And so we inject the drugs right before the encoding a one-shot experience, so right before familiarization. And we check again on the next day if there is an impairment on the learning. And so here are the graphs for the CB1 receptor blockade. As you see again, the blockade of the CB1 receptor alter again the one-shot learning, but not so much the 2-shot kind of learning part. And for the NMDA receptor blockade, we actually don't see a significant impairment in the task. So it doesn't seem really required for learning the task at all, actually, in this dorsolateral striatum. And so just to kind of finish, I would like also to give you a sense of another question that we explored in this project, which is how we can make predictions basically of the plasticity, about the plasticity rules that occur in vivo based on the activity pattern that are actually occurring in vivo, like that during the task. And so we did this work in collaboration with a team in Lyon, based on some neuropixel recordings that I did during the sticky tape task. And so the idea was to do simultaneous recordings of the cortex and the striatum acutely. So we put the mice in this kind of mobile home cage design, where they could still encounter and the tape and get stuck to it. And then we kind of measured, just to give to get an order of magnitude of what activity was going on in the network, like just the number of spikes that were actually happening during the contact, and see whether or not it would kind of match like the like the in vitro estimate that we used, like for eliciting like the eCB-LTP. And so if you look at what's going on for short contacts here in pink versus like the long contacts in green, what you see is that actually there are a lot of neurons that remain kind of silent during short contacts. So that's like the graphs here with the like number of spikes, like the distribution of the neuron with the at least X spikes. And only like about like 30% of them fire like 2 to 20 spikes, so which is like the window that is used in vitro like to elicit the eCB-LTP, but that fraction is very much smaller, like if you look for the longer contact, which is kind of expected again due to the duration of the contact. But and we can also be a bit more precise by counting not just the number of spikes, but the number of pairings, which is really what matters if we think of spike timing dependent plasticity. And so we don't. So we don't. And so in this approach, we were like very like inclusive. So we looked at every possible pairs. Yeah.
I think you might have recorded from that same number of neurons as the total number of inputs a striatal cell would get.
So probably not. Again, like a striatal neuron like receives a lot more inputs. Like there is this kind of tunneling of information, but
so this would be number.
Yeah.
I think the practice bears.
And exactly, I was going to say that we also looked at a more specific subset, which shows higher correlation during the contact.
Check this rotation.
Yeah.
We can have a look at the data, trying to identify connected pairs
From the cross correlation.
Yeah, and
Yeah.
it's not sure but yeah, we have this tolerance.
And it makes a lot of sense that it doesn't necessarily work, right? Because for MSN, like a striatal neuron to fire, it needs quite a lot of inputs. So you wouldn't necessarily easily identify it from in vivo recordings, right?
If you just assume random sampling and you knew the number of inputs and you knew the number of samples,
Yeah.
We do
Yeah.
but it's completely flooded because of because you need 10,000 coincidence spikes in the cortex to generate Striatal spike. If you add to the use of correlation, but keep the problem into the nose. Essentially.
Yeah. But so even being like very inclusive, like we could still observe that there's like a non significant percentage actually of pairs that show like levels of activity that could match the eCB-LTP like window. And overall, like the levels of activity for the short contact is relatively sparse. So that's kind of the big message here compared to what you see for long contact, in which like the correlations in activity is like much stronger and like repeated a lot more, like faces like a lot more repetition. So that makes it again like the short contacts much better likely candidate for having this eCB-LTP induction events. And so to kind of test this hypothesis a little bit more precisely, well, so two colleagues in Lyon developed like this mathematical model like of cortical striatal STDP. So it basically describes like the kind of kinetics of the main molecules and receptors that are involved in the induction of the eCB-LTP and also LTD pathways as a function of the spiking inputs, basically. So it's very detailed model, so based especially on the calcium kind of thresholds. And so it was validated in vitro and then adapted a little bit to the in vivo firing rates. And so the main determinant, as we discussed just before, is that the induction of the eCB-LTP depends on CB1 receptor activation, which kind of translates to the calcium kind of levels. And so LTP will be like evoked for higher like levels of endocannabinoids and LTD for lower levels. And so we use that model and the spike train inputs that we recorded in vivo to then estimate the level of CB1 receptor activation and the like subsequent kind of eCB-LTP induction events. And so I'll give you an example here of one trace that we can generate for one animal. So over time, basically what you see is like the eCB-LTP induction events that can occur with the natural activity going on during the recording. And so what you see is that if we correlate it to what's going on in terms of behavior, is that there is a big peak here that is typically, the contact with the sticky tape. And there are other moments also where you see some peaks, which we could correlate with some other behaviorally like kind of relevant moment, like the sniffing with a tape or like grooming events. And if we know kind of try to generalize to like all the animals that we recorded from, what we found is that in mice, especially like experiencing a short contacts, which are like the top 4 mice, we could see a much higher density of eCB-LTP events predicted by the model, like happening during that period of contact with the tape. While for the longer contact, there was still like, again, like some like relatively high probability to induce some LTP events, but those were like much less like densely like kind of packed over like the duration of contacts.
Isn't this confusing because for the first, say, 10 seconds of the long contact, there's no difference between the long contact and the short contact. So why wouldn't the events be the same?
Well,
to the first second?
We don't really know if there is no difference in the mind of the mouse even at the very beginning, right? Like the stress associated even to just like the contact, the very initial contact might be different. And so the way the mouse reacts might already be different at the very initial seconds, right? Like, so
how long will be determined by the mouse states?
I think like there could be a lot of determinants that that might be involved, and we actually saw a correlation between like the glucocorticoid levels, like post like familiarization, and the avoidance like index. So I think like the emotional state and like attention and arousal maybe of the mice could definitely like also be important parameters even. And that could actually be an interesting prediction too, right? Like to see if that's actually, and maybe like if you look at like mice, the mouse 8, right? Like there is maybe less difference compared to the other ones, and like try to correlate it to like even like other neuromodulator that we could measure, like see if it could be so just to wrap up, I think it's kind of time. I don't know how, so we use kind of different approach to kind of that somehow converge again to highlight this critical role of the endocannabinoid system and of the eCB-LTP to kind of mediate this one-shot learning in the sticky tape task. So kind of to put all the pieces together again, we have this kind of schematic where we see like the in vivo LTP for the brief contact and for the learners, basically, where we don't for those that don't learn. And then we do this like overall manipulation where we can actually get some sense of causality maybe. And I think one obvious question is also how like to what extent do these results somehow generalize, right, to other tasks maybe? And so we started just exploring this, and actually these experiments were among the very first one that I did during my PhD. So it's kind of the story being a little reversed here. But so I initially looked at the very first trials of the accelerating rotor rod. And so this is very classical kind of motor learning task, which typically takes time for the mice to acquire with like very fast learning kind of motor state, like fast learning stage followed by a slower like refinement of the motor pattern. And so I kind of tried to stretch that very fast initial learning stage by having just 2 trials per day. Well, usually it's like 10 trials per day, more like extensive training. And try to see whether or not like the eCB-LTP could actually also be involved here. And so what I did was the same kind of occlusion experiment that I described for the one-shot learning test. And what I found was also some sort of occlusion here for the early stages of this task. And later on, so we also tested the D2 LOXP mice, and we also found like a drop in performance that was actually significant in the very first stages of learning. Again, like few animals, so like few, like, you know, kind of initial investigation, but I think that's a kind of important question also to test later on. And with that, I'd like to to thank you for your attention, and to thank like the 2 PhD labs. So Laurent Venance in Paris and Jonathan here, and also the collaborators in Lyon. Yeah, thank you. Questions?
I don't want to jump in front of everyone. So a weird prediction from a plasticity rule, right? There's this range of pairings between where the ECB mechanism is now depressing spot and before the NMDA one kicks in.
Yeah.
Would that suggest that, you know, somewhere between like 20 seconds and 2 minutes in the sticky tape, you're in this region where you don't learn? So that like there could be an intermediate range where, you know, a short bad period is good to figure out, a long bad period, or a medium one, you don't learn, and maybe weird. It's a lot of like the biochemistry suggests.
Well, yes and no, actually, because in vitro, like what we found also is that there could be like mixed plasticity, like especially if you were modifying the firing rate, like the rate of the stimulation, there could be mixed contribution of the ECB and the NMDA, like so I think at some point the 2 systems somehow
So
like become activated.
You never get the precise 10 millisecond people's continuing.
Yeah, I think it would be hard to just. Yeah, I think it's more of a continuum.
So
But,
I guess, you know, so back many years ago, we started this project, we had to say, yeah, maybe, you know, this kind of silent period was the time you know, you want to be able to have fast learning and learn from a single experience. You also want to not become superstitious, you know, and just associate, you know, your plan to a good grade and stuff, you know. So as the experience repeats, you want to be able to refine your learning. So maybe that's a feature that you're not stuck with something you learn immediately and then revert to. So we thought this could be actually useful. What I think, I mean, the reason why I don't think you don't learn if you just touch a little too long but not long enough is that the observations that are made for this specific are really in vitro. Okay, so the only essentially there's very little background. There's very little reverberation. So there's no transfer to. I mean, it's a slice. Which there's very little activity. And there's a constant bath of the, I mean, I don't want to
Yeah, no.
say that there's a constant bath of the pending, so you also lose this bath of the learning. So what you see is actually one of the building blocks of the learning, which is this ability to activate very fast, relying on endocannabinoids,
Yeah.
but it's not necessarily exactly what happens in vitro. Just a possibility.
I guess the second question, have you thought about replay? And I noticed like the NMDA learning was like stronger day labeling on face than 30 minutes later.
Yeah.
And do you think that's like reactivation? And is there any evidence in this system for reactivation?
Yeah, that could be like, and I mean that's actually what the next PhD student is kind of trying to investigate right now. So I don't really know exactly what he found yet, but yes, that's definitely like, yeah, like the NMDA component and the consolidation part. That's definitely a question that we wanted to look more closely. And yeah, you're right with the occlusion that it's actually stronger and more clear at 24 hours later. So yeah, that would make sense that the replay, and it's been described in the striatum too, right? That
Question that I should know is
Yeah.
related to that. When you do slides,
Yeah.
you're pairing. So I mean, Jackie here has like been looking at whether you could have rather plateau activity after the with I was wondering perhaps to the slices, when you do a pairing after an activation, you don't have your reverberation, so it's just what you
Yeah, because we keep the neuron also in a very non-depolarized state, right? So I guess we also block the possibility that there could be any reverberation.
It's ratings only, I think it's never, it's like 75.
in terms of cost. I mean the cost things
Right, so you can have cortical memory.
and
Right.
so with the neuropixels data, there's phosphates and striatal. To do that, but also with that ongoing activity, cortex can also drive the striatal.
Yeah.
So even if the cortex is striatal, yes, so that's a fundamental difference also.
Yeah, I mean, you only look at just also one direction, right? Like you don't have the full loop. Yeah. Well, thank you. Yeah.